In this tutorial, we will be demonstrating how to prepare a protein-loaded membrane for use in Western Blot immunoassays using the semi-dry transfer method. Western blots can be used to detect specific proteins within a sample, as well as information on the relative size and abundance of the protein. First, determine the appropriate type of gel and running buffer to use for the sample you wish to test. Different gel types and running buffers will affect the size distribution of the protein across the gel. Choose a system where your protein of interest will be centrally located and not along the edge of the membrane. Place the gel cassette in the electrophoresis box with the running buffer. Make sure to add enough running buffer so that the wells are completely submerged. Add a molecular weight marker and the sample supplemented with the loading buffer to the proper wells. Connect the box to the power source with cathode to cathode and anode to anode. Start the flow of electricity according to the specifications of your gel and electrophoresis system. Once the sample has reached the end of the gel, turn off the power source and remove the gel cassette from the electrophoresis box. Carefully remove the gel from the gel cassette. Avoid creating any tears or rips in the gel. Cut off any unwanted parts of the gel, such as the wells or extra gel at the bottom. Rinse the gel with water and briefly equilibrate the gel in blotting buffer. Soak 4 pieces of blot filter paper and 1 nitrocellulose membrane in blotting buffer. Alternatively, you can use a PVDF membrane, which requires an activation in methanol for 5 minutes. Place 2 of the blot filters onto the anode of the transfer system. Use a device to roll out any air bubbles between the filters. Next, place the membrane on top of the filters. Avoid creating any air bubbles between the filters and the membrane. Then, carefully place the gel onto the membrane. Avoid creating any air bubbles between the membrane and the gel, and try not to move the gel once it has been placed onto the membrane, as proteins will immediately start to transfer. Finally, place the last 2 blot filters on top of the membrane. Again, use the device to roll out any air bubbles between the filters. Finally, close the transfer system and apply weight to the system to further prevent the formation of air bubbles. Connect the transfer system to the power source with cathode to cathode and anode to anode. Begin the transfer using the voltage and time length specified by your blot transfer system. Once the transfer is complete, turn off the power source and open the transfer system. Remove the membrane, rinse it in water, and place it in Ponceau S solution to check the efficiency of the transfer. If desired, the gel can also be placed in Kumasi solution to check the efficiency of the transfer. Note any air bubbles on the membrane before using in a Western blot.